And if you put higher pressure through these tiny capillaries, what might now get pushed across the wall? Blood cells, yeah. Blood cells like red blood cells, even proteins can escape through those capillary walls. So that's yet one more reason why hypertension or high blood pressure can be kidney damaging, because it can push blood cells and proteins across the wall. Once you push them through the walls of the capillaries, you can't get them back. They're gone. So it's, something we can test for in your urine with those urinalysis strips, or, a urinalysis, a more fancy system. But hey, if it's gone, it's gone. You can't reclaim those items back. We have this thing down below. The next bullet point is called the glomerular filtration rate, or GFR for short. And this simply is a volume of filtrate formed by each kidney each minute. It's kind of hard to say, okay, pee every single minute, right? That's kind of impossible to measure. So what we do is we measure this over a 24-hour period of time. That gives us a more accurate look at how well the kidneys are functioning. So even though it tends to be related to simply a minute's period of time, the measurement is done over a 24-hour period of time. Notice that in adults, the GFR in both kidneys is around 120 mils per minute. 120 mils, that's about a third of a Coke can. That's not a whole lot, but hey, that's a pretty efficient process. And if you do the math here, I've got a question that says to you, how many liters of proteins, or plasma are filtered each day? If you do the math, that means that we can filter about 180 liters of plasma per day. How much blood do you have anyway?
5 liters.
About 5 liters, maybe 6 if you're a larger person. And of that blood, say if you've got 5 liters, how much of that is actually plasma? Half of it, exactly. About half of it is plasma. Boy, what a machine your kidneys are, right? Because if you consider you have around 2.5 to 3 liters of just plasma, I'm not talking blood cells right now, talk about filtering that some, 30, 40 times a day, 50, 60 perhaps even. That's a tremendous job that your kidneys are doing in filtering that blood. Notice that out of those huge volumes of plasma that we're actually filtering over and over again every day, we actually only lose about 1.5 liters of fluid. Only about 1.5 liters of urine are excreted from our bodies in a day's time. So what an efficient process by which we reclaim what we need to keep and let go of what we don't want to keep. There's another measurement down there called renal clearance, and that is another way to measure how effective the kidneys are doing their filtration job. There's a little carbohydrate called inulin. You'll notice it's down there at the bottom. This is a plant polysaccharide that is used because this is something that we tend to filter and we don't tend to reclaim or reabsorb. So where does this inulin eventually end up? In your urine. Yeah, so it's a way to say this is how effectively the kidneys will clear, there's your word clearance, certain items out of the bloodstream and remove them into the urine. What if you have a low renal clearance? What does that tell us about kidney function? It's poor, right? So the kidneys may not be doing their job as effectively. So things like inulin can be given by way of blood, say for instance. We can also use creatinine, which is a muscle storage device for ATP. That can also be used to detect renal clearance. All right, so far we have the glomerulus as the site of filtration. What is found in filtrate? Good. Plasma, minus the proteins. What about the blood cells? No, keep them away from the filtrate as well. Filtrate forms as long as blood pressure is normal. Well, what happens if you've got low blood pressure? Angiotensin, good. What does angiotensin constrict? Efferent, good. It makes sure that the pressure here is high. Why would it make better sense to constrict the efferent arteriole and not the afferent arteriole?
It'll build pressure in the glomerulus.
Right, because if you have high pressure going in, it doesn't guarantee that you're going to have high pressure going out. So this maintains the pressure in the capillaries by having that, angiotensin constrict those efferents. So there we're going in. There we're going out. Once you actually filter out plasma from the blood, we're going to capture it in a little sink bowl, and that little sink bowl is called the Bowman's capsule. The Bowman's capsule surrounds this glomerulus. The alternate name for Bowman's capsule is glomerular capsule. So we're just going to capture all this filtrate in this little bowl surrounding your glomerulus. So it's like having a wet, dirty dishcloth, wringing it out in the sink, and then whatever's wrung out in the sink now travels down through all the pipes of your house. And that's where we're going next. These two structures together, the capsule and the glomerulus, are known as the renal corpuscle. So now we're going to travel down the renal tubule portion that includes PCT, the lupus or nephron loop, and then of course the BCT. What's the difference between PCT and BCT? Yes, where it is, because of course PCT stands for proximal convoluted tubule. Proximal convoluted tubule means we're close to something. What is our point of reference here?
Glomerulus.
Glomerulus, yeah. So PCT means simply we're close to the glomerulus. BCT means we're far away from that glomerulus. So we're going to look at the PCT right now, and that's where we're going to start the process of tubular reabsorption. That's found on page 115. And information in your textbook is page 849. Probably an entire course doing all the urinary system. If you've read through the textbook, you see that they go into lots and lots of detail. So even though there are activities that occur there, like secretion, we're going to stick with the highlights. So keep that in mind if you happen to go back and read the book, because it is chock full of information. All right, so the PCT is one of the locations for reabsorption. Back in chapter 19, we talked about reabsorption occurring at the venous end of the capillary. So this time, we're talking about reabsorption occurring from the PCT into a set of capillaries that surrounds this whole structure. You'll notice that as we leave the PCT, we drop down into this descending limb of the lupus Henle or nephron loop. We come back up through what? Ascending limb. It gets a little wider up here, and then we start the wigglies again when we travel through the T or the DCT runner. And then what's the final portion of this whole renal tubule called? Collecting duct. The reason I want to show you the whole entire thing is because traveling across, wrapping around all of these tubules are teeny tiny capillaries called peritubular capillaries. And these peritubular capillaries crisscross and wrap through all of these tubes. Okay? And as you might expect, these capillaries are under fairly low pressure. So what's easy to occur under low pressure? Well, yeah, reabsorption can easily occur under low pressure. So any items that you may have in your PCT, lupus Henle, or DCT, if you wish to reclaim those back into the bloodstream, the items are moved into the capillaries. Okay, so we've got the peritubular capillaries wrapping around all of this renal tubule. So let's do the PCT then, here on page 115.